Mr. Michael Bush.
Thanks. Is this working? Okay. I'm not sure I'm the leading expert on top bar hives. I think Sam's got way more than I do. But I've probably had him longer than he did. But I think he may be more of an expert on top bars than me. But anyway, I'm going to talk about 4 simple steps to healthier bees. And you're going to find a lot of this is counterintuitive. But I think you're going to find in life things are often counterintuitive. You know, if you want to maximize the mosquito population in this area, you know how to do that? You go spray for mosquitoes. Because you'll kill off all the predators that eat the mosquitoes, and the mosquitoes will bounce back so fast that you won't even notice the difference. There'll be less mosquitoes for like 2 days, and then the new ones will be coming out, and then all the dragonflies that would have eaten them are dead. All the bats that would have eaten them are either dead or they're starving. You know, you've just put a major dent in all the predators that will eat the mosquitoes, and now you have more mosquitoes. So if you want to maximize the number of mosquitoes, you go spray for mosquitoes. Now the problem is, as human beings, we tend to see things as direct one-to-one relationships. We think, well, I don't want as many mosquitoes, so therefore I'm going to kill the mosquitoes. And we don't take into account that that's how you get more mosquitoes. That's not how you get less mosquitoes. It's where I live, prairie dogs were the scourge they were trying to solve. And so how did they try to solve it? They poisoned them. And what did they do? They killed all the predators that killed the prairie dogs. And what did they end up with? More prairie dogs. That's how life works. Reality of life is that everything's interconnected. And if you don't take those connections into account, you just end up with the opposite result of what you were trying to do. So I'm going to give you some things that I think are common sense choices, but they probably seem counterintuitive at first. Before you take too many notes, I just want you to know that these presentations, all the presentations I'm going to do are all out on my website. You can go look at them anytime you want. You can still take notes if you want. I just want to save you the work. If you'd rather just, you know, if it's something you want to refer back to that's in this presentation, you can go look at it later. Also, I'd like to say, I'm going to sell some books over here, but everything that's in my book is on my website. You can read it for free. People just kept bugging me to put it in a book, so I kind of accidentally wrote a book. I kept putting answers to things that required more detailed answers than just yes or no on my website, so I didn't have to keep repeating myself on the bee forums, and then it kind of grew and it grew and it grew, and people kept saying I should write a book, and so I organized all of that into a book. There's really nothing new in the book that isn't on the website, but if you want a book, it's organized differently. And maybe polished a little more, but it's the same information. So, my first step to 4 simple steps to healthier bees is no treatments. That obviously is back to my counterintuitive stuff. But here's why. I used to say there were 50 kinds of mites on bees. I did some more research and found there's 170 of them. There's 170 different kinds of mites that live on bees, and if you go to that link there, you can get an entire list of all 170 of them that normally live on bees. And of all of those mites, there's like 3 of them that cause a problem. One of them we don't have yet, and the other 2 are the varroa and the tracheal mites. And the rest of them, they're either benign or beneficial. Nobody really knows because nobody's really studied them. They just know they live on bees. They don't seem to do any harm, but they don't really know what they do. But there's probably 30 kinds of insects. That's an estimate on my part, having taken a lot of bees out of trees and looking at the detritus in the bottom and the different kinds of debris beetles and the different kinds of ants and wax moths and whatever that's living in the bottom of the hive. There's all kinds of insects that live in there. There's probably a lot of mites that are just debris mites that eat the debris that don't even live on the bees. But there's 8,000 microorganisms that they've actually identified. Now these are organisms that they could actually culture in a petri dish. This isn't actually how many different organisms live in a beehive because they can't culture a lot of them. They're doing a lot of research recently because now they can do they can do the DNA stuff now, so they can try to identify new organisms. The problem is when you break down the DNA of what's in a beehive, it kind of all gets muddled up, so it's hard to distinguish, did this come from a different organism or the same organism? And but they can culture in a petri dish. There's 8,000 things. There's 8,000 microorganisms living in a beehive normally. And there's a reference to a USDA article that quotes that. I think that number comes from Martha Gilliam's research. So there's the 8,000, the reference to the 8,000, but there's Martha Gilliam's research if you want to go look at some of her research on specifically what some of those are and what the benefits of some of those microorganisms are in a bee colony. So what's the effects of treating on that ecology? Fumidil, which is a common treatment people use to treat for nosema, it kills mostly yeast and fungus, but it probably kills some of the bacteria too. It's an antibiotic. Terramycin kills mostly the bacteria, and it's an antibiotic. The new antibiotic that's the in thing everybody's using is tylosin, which technically they're not supposed to use unless they can prove they've got terramycin-resistant AFB, but the commercial guys are buying it by the ton and just putting it in their hives without any concern for that. But it's a broad-spectrum antibiotic that's longer lived than the tetracycline, and none of the microorganisms in the colonies have built up a resistance to it, so it's pretty devastating to the whole ecology of the microbes in the hive. Essential oils are pretty broad spectrum. They kill everything from viruses to bacteria to yeast to fungus. They basically, essential oils are the defense mechanism of the plant. You're basically taking the sap out of the plant, distilling out the stuff that the plant uses to defend itself against microorganisms. And if you put that in the colony, you're killing off a broad spectrum of microbes again. Organic acids mostly kill microbes by shifting the pH dramatically. You've shifted the pH dramatically, and then certain bacteria, almost all the bacteria can't live in that kind of a pH, but what few that can are entirely different than the ones that would have lived otherwise. It also kills off those mites, and anything you're using to try to kill varroa mites is going to kill off all the other mites. So you're killing off all of those and probably some of the insects. Then acaricides, I'm lumping the stuff that used to be called insecticides that they relabeled as acaricides when they sold it to beekeepers because somehow they didn't think it would work to call it an insecticide. But basically they took organophosphates, which is an insecticide, and tried to use it to kill a bug on a bug. And those are going to kill, of course, all the mites and most of the insects and probably all of your bees. But by that I mean the systemic stuff like checkmite, apistan, those the new one what's the name of the new one they just came out with that's an Amatraz product? I don't remember. Yeah, it's all Apa something. Yeah, anyway, they just came out with an Amatraz one. Of course, all the commercial guys have been using the Amatraz for decades, but anyway, those that's what I would call the acaricides. But this is part of the problem with defining what a beneficial organism is that a lot of times, well, let's just take your skin for example. You have bacteria that lives on your skin, and if it wasn't living on your skin, then fungus would live on your skin. So you need this Staphylococcus aureus that lives on your skin in order to defend you against the fungus that would live on your skin otherwise. So if you kill off the bacteria on your skin, you're at risk for a fungal infection. If you get on some really heavy-duty antibiotics, often the doctors will put you on an antifungal even though you don't have a fungus infection because they know as soon as they kill off all the bacteria, you're liable to get a fungus infection. And so they're trying to head them both off. The fact is, even what sometimes is called a pathogen, you get Staphylococcus aureus in a cut and it starts growing, it's a pathogen, it'll kill you. But it's not when it's living on your skin. So a lot of times it's a question of balance. A lot of times things we think of as pathogens are actually beneficial as long as they don't get out of balance. We know chalkbird spores prevent EFB as long as the colony can maintain a high enough temperature and as long as the humidity isn't way too high for too long. Chalkbird hardly ever gets going in a colony. But the spores that are there will help prevent EFB even though you're not getting chalkbird. We know there's bacteria that crowd out EFB and AFB, not only in the colony in general, but there's bacteria that live in the gut of the bee that creates an environment so that the AFB and the EFB tends to die in their stomach so it doesn't get fed to the larva so that it doesn't spread. And I'll reference that research somewhere in here. If I don't, remind me, I'll look it up for you. Stone brood toxin actually is what they use to make fumidil. The fungus that causes stone brood is cultured, and they gather the toxin and they sell it to you as fumidil. Or fumigillin, or whatever they're calling it today. They keep changing the name. I don't know if that's, in theory, that's because they keep changing the delivery mechanism so that it'll dissolve better, but it's the same active ingredient. So I mentioned this kind of on the EFB, AFB one, but there's flora in the gut that actually creates a film. It creates a biofilm. The bacteria, or a colony, and this colony coats the whole inside of the gut of the bee and protects their gut from things like nosema and from things like EFB and AFB. It kills off that AFB and EFB because of the acidic environment in their stomach, and it can't do well. It can't reproduce well in that environment. There are yeasts and bacteria that are essential to the formation of bee bread. Probably most of you think that bees eat pollen. Actually, bees don't eat pollen. Bees can't digest pollen. They can collect pollen, they can ferment pollen, and they can eat bee bread, and they can feed the young with what they secrete because of the bee bread, but they actually can't digest pollen. Pollen has this hard capsule on it that they can't get through. So it's essential for them to be able to ferment this into bee bread. And if you put antibiotics in there, you kill off all the bacteria that's required for that. And if you put fumidil in there, you kill off all the yeast that's required for that. And you're going to end up with malnourished baby bees because they can't digest the pollen, and therefore they can't feed their young very well. This is the study. If you just search on symbionts as major modulators of insect health, I think you'll find this study online. You can read the entire study. It's published. You don't have to have any special subscription to get to it, and it's about that biofilm, and it's about the fact that the antibiotics disrupt that biofilm, and that even feeding sugar syrup disrupts that biofilm, which Martha Gilliam also talked about, that the sugar syrup would disrupt it. So we kind of already talked about the benign and the pathogen thing, and that's that even organisms that we don't really know what their benefit is, they may be beneficial just because they crowd out things that aren't. They fill a niche in the ecology that otherwise would get filled with something else that may not be what's supposed to be there. It's hard to describe this, but there should be some easy thing in our lives that would correlate. But in an environment like a bee colony, or even in your gut, you have thousands of different kinds of bacteria living in there, and they're all kind of in a balance where this one actually creates a byproduct that this one lives on, and this one actually creates a byproduct that suppresses this one from taking over, and this one actually kind of barely survives, but it makes a little bit of a byproduct that feeds this one over here, and then and suppresses this one over here. And so there's this really complex balance of all of these organisms. And a bee colony is the same way as your gut. It's got literally 8,000 or more microbes that are living in this colony that are all in a balance. And anything you do that upsets that balance is liable to cause one thing to overgrow that shouldn't. And you've got something that essentially you think is benign or doesn't do anything when actually it's doing something beneficial. The other thing about not treating is it puts the selective pressure where it belongs. I can't believe how often people want to argue about this, but it seems rather obvious to me. If you want bees who can survive without treatment, you have to stop treating them. Because otherwise, how do you even know if they can survive without treatments? The reality is their survival is dependent on such a complex system that I don't think you can isolate it down to this one thing and say, well, I measured this one thing, and this didn't change this one thing, or this did change this one thing, and so therefore I think now they could live without treatments. No, the only way to find out if they can live without treatments is to stop treating them, and the only way to choose the ones who can live without treatments is to pick the ones that survive when you don't treat them. And it doesn't make any sense to me to even argue about that. I don't know what, that seems intuitively obvious. But you put the selective pressure on the bees where it should be when you stop treating. As long as you treat, you don't allow any selective pressure for the things that they're facing. You know, you've got AFB, EFB, varroa mites, tracheal mites, nosema. Those are the big ones that most people are concerned about. And there's no selective pressure for them to be able to handle these things as long as you keep treating them for these things. Plus, we just talked about how you're treating them for them. You've disrupted the very mechanisms that protect them from it in the first place, which is the bacteria in their gut. So I don't see how you can breed resistance to those things until you stop treating them. Anytime you do treat for anything, you put selective pressure on the parasite or the pathogen. Let's take varroa mites as an example. If I'm raising bees and I'm treating for varroa mites, the only varroa mites that survive are the ones that can reproduce fast enough that I can't kill them all. That's basically what's going to reproduce, what's going to survive. In the case of if I'm not treating, the ones that survive are the ones that don't kill their host. The ones that kill their host don't survive because they kill their host. Well, you can argue in an apiary they might, because they'll get robbed out and get moved next door. But the fact is I keep breeding more virulent mites by treating, because I keep reproducing. It's the ones that reproduce fast enough to keep up with my treatments and resist my treatments that manage to survive. So I keep putting selective pressure on the parasite to be a super parasite while not putting any selective pressure on the bees, so I get wimpy bees. So I got wimpy bees and super parasites. That's the result of treating. And I don't see any way around that except to stop treating, and then you can get parasites that are in a balance with their host and bees that can handle it. So as long as you keep creating this artificial system and you keep propping it up and pushing it in different directions with your treatments, you create a whole different system. You know, the one encouraging thing I'm starting to see come out of this whole CCD thing is that the scientists are finally starting to view a colony as a system. Up until now, they've almost consistently, in the whole time I've been keeping bees, and I started in '74, and I read stuff, old stuff, because it was what was in the library when I started, you know, from way back. And the scientists have always viewed this as this one pathogen, this one smoking gun. We're going to kill this pathogen, and everything will be all right. And they've never viewed it as a system. And now they're finally having to view it as a system because they can't put their finger on the smoking gun. They can't find that one pathogen that's the cause of their problem. And they're finally having to say, well, you know what? We have a whole complicated system, and we've been messing with this system so much that it's hard to put your finger on which part of the system that we broke to cause this to happen. So I think as long as you keep propping it up and you shape the interactions of what's going on by what you put in there. You shape what bacteria flourishes and what ones die, and what one, you know, and you've changed the whole system into something else. As long as you keep doing that, you really can't breed for bees that can live without that. So my third reason to not treat is to keep the combs clean. This is some foundationless comb here. That's a foundationless frame. There was no foundation on there. It's all ugly and crooked like they told you it would be, right? I won't say they don't ever do ugly, crooked comb, but I've had them do it on plastic foundation just as much as I've had them do it on foundationless. So the thing you got to understand about most of the chemicals that people are sticking in the hive is a lot of them are lipophilic. That means they're basically oil-based. They're attracted to oil. They go into the wax, which is basically a very high viscosity oil, and it stays there. It doesn't go anywhere. And what breaks down most of them, the fluvalinate, which is the apistan, and the checkmite, which is an organophosphate, the coumaphos, both of those really only break down in ultraviolet light. And there's no ultraviolet light in the colony, you know. So it's just building up. It's not going anywhere. So the fourth reason is, and some of this applies to different chemicals, like the keeping the combs clean the combs clean is mostly about the amitraz and the coumaphos and the fluvalinate, which would be the checkmite, the Apistan and whatever they're calling the new Amatraz one, I forget. But Apigard or whatever it is. The other set of chemicals that people are typically using is essential oils, and this probably applies more to the essential oils than the acaricides. But the communication in the hive is pretty much by smell. I mean, we all know the dance language thing. You know, the bees come back and they do their little dance, which, by the way, is happening in total darkness. So obviously, it's the vibration that they're feeling, and they're closely following. If you've ever watched this in an observation hive, they closely follow that bee because they're feeling the vibration. They need to be close enough to get their antenna out there and kind of feel what the bee's doing and feel the vibration on the comb and whatever. But other than that, almost everything else that goes on in the hive, all the checks and balances, all the feedback mechanisms that cause everything to work smoothly are all based on smells. But between the dance and recruitment and smells, that's pretty much everything that's going on in the colony. So you've got, and usually there's a kind of a combination of the two going on, because when a forager comes back and does their little dance, they share some of that nectar so that they know what it smells like. So they know what they're looking for. They know, okay, I'm going to go that way, that far, and it smells like this. And they go that way and start going that far and start smelling with their antenna to try and find that flower. And they do. So it's a combination of smell and the dance. And then the feedback mechanisms that are going on, the pheromones from the open brood cause them to want to go get more pollen because they have to feed the brood. And they need to smell that pheromone in order to know to go get more pollen in order to feed the brood. And it's true, there's also recruitment going on. There's a pollen collector who comes back and says, here, you want get some of this. It's over there and it's this far. But it still comes back to that smell of that pheromone that's part of what's driving this thing. So smell is it's an essential part of how the colony communicates. It's how they know there's a queen. It's how they know the brood needs to be fed. It's how they know they need to go get more pollen. All these things are driven by smell. And all these essential oils not only interfere with that sense of smell, but quite a few of them actually mimic some of those pheromones. The lemongrass oil that's in your Honeybee Healthy is almost exactly the same as Nasinoff, which is sort of the get-together pheromone. It's this is where we are. We all need to get organized and figure out what we're doing. That's what that pheromone is. If you want to see them do it, just take the lid off a hive and pick up a frame and shake the bees back in and put the frame back in, and you'll see a whole bunch of bees come up to the top, stick their butt in the air, and start fanning. And that's Nasinoff pheromone. In fact, if you're planning on moving your hives and you want everybody to come home, just do that and then wait about 30 minutes and almost everybody will be home, because once they start doing that, it tends to kind of build. More of them will come back and start doing it, and pretty soon they're all home, and then they're like, okay, what are we doing now? And then, oh, well, I guess nothing's going on. I guess I'll go back out and start foraging. So, but that's what that smell is. Yeah.
When you smoke the hive, you're blocking the receptors on their antennae. How long will those receptors be blocked?
Well, anybody who's worked a hot hive has a pretty good idea how long that is, don't you? I mean, it's good. It's good for maybe 5 or 10 minutes, and then it obviously in 5 or 10 minutes they're starting to act like you never smoked them. So I would say that's not too hard to estimate. I don't know, but it doesn't seem to last long. And it does interfere with their sense of smell, and that's why it works. But it doesn't seem to interfere with it for very long. Essential oils, on the other hand, you feed a whole bunch of syrup with wintergreen in it or whatever, you can smell wintergreen there for the next 3 weeks. So I don't know how long that interferes. Probably a lot longer. Of course, I can still smell smoke maybe the next day.
Is that part of the idea behind using menthol-based products to treat for nosema and other things, tracheal mites?
Menthol is mostly for tracheal mites, and it's because it kills them. But the theory of the grease patties is that it confuses the mites because they can't smell whether it's a young bee or an old bee, and they have to find a young bee because they can't infest an old bee. There seems to be a lot of not specific information about how why they need a young bee, but I suspect it's because the spherical actually is small enough. I think that the which is what they breathe through. I think that's small enough the trachea might actually has trouble getting in there, but there's plenty of room in the trachea once you get in, you know. So I think they have to have a young bee because that chitin around that is soft enough that they can chew their way in, and an older bee they can't. That's my theory, but I don't know. I've never really seen a lot of research on it. All they know is it works, and they suspect that the reason that the grease patties work is it confuses them and they can't tell a young bee from an old bee. But I don't. Menthol isn't really working on smell. It just works on the fact that it kills the mites. So. One of the arguments everybody always gives is, you know, if you stop treating, some of your hives are going to die. Well, what I'm finding all the time is everywhere I go, I find people who are treating and all their hives died, and so they just quit doing it and they started raising bees not treating, and they're having much better luck. So I don't see any evidence that more of them are going to die if you don't treat them than are if you do treat them. The last Bee Informed survey basically came up with the winter losses for people who were treating was 30%, and the winter losses for the people who weren't treating was 30%. So I don't know that you can prove that any more or less of them are going to die if you don't treat. But assuming some more of them died, you can always look at it that's actually the ones that should have died. You've actually headed the gene pool in the right direction, which you couldn't do when you were treating. If they die, you get rid of the bad genes. If they survive, you got the good genes. The thing about beekeeping, whether you're treating or not, I think you need to try to get more self-sufficient, and I think you need to do splits and plan on what your losses are. If you know you're going to have 30% winter losses, well then let's go into winter with 30% more bees than I want in the spring, and I'll have the bees I want in the spring. And then I don't have to buy packages, and I don't have to keep bringing in genetics from somewhere else that aren't really adapted to my climate. I know here they're going to preach to you that AHB are horrible, and if you talk to all the people who are collecting the wild swarms around here, I think you'll find that they're not as bad as you think. I think you'll find that some of them are as bad as you think, but not all of them. In fact, most of them are not. And I think really if I lived in an AHB area, I would be trying to breed local bees that were gentle. And if everybody would be trying to breed local bees that were gentle, you'd be doing what people have been doing for thousands of years successfully, which is breeding local bees that are gentle. And that's how we got the gentle Italians that we brought over here in the first place, was people in Europe had been doing that for thousands of years. We just need to keep doing it. I don't think it's that complicated. And honestly, my experience is the meanest bees you meet are the hybrids. The meanest ones you meet are when you bought that nice new Italian queen that you got from wherever, and you put it in that hive and it crossbred with whatever is out there, and those are the mean bees. The local ones that aren't being crossbred are actually not that bad. But that's my experience from having gone around the country to places like this and talking to people. Yeah.
Actually, last night we heard that the state of Florida is planning on actually requiring beekeepers to recruit like every 6 months.
You want to know what I think the result will be? I think you'll have more bee attacks and more mean bees. That's what you'll accomplish, because you'll keep bringing in purebred Italians who will keep crossbreeding with the locals, and those crossbreeds are always the ones that are mean. And so I think you'll end up with more mean bees if you do that than you will if you try to breed local bees that are gentle. It's another one of those, you're not looking at the next step. You're assuming that's going to accomplish that everybody always has gentle Italian stock, and that's not what's going to happen, because they're going to supersede. They're going to.
Bees out of Texas doing for you, sir?
I got some from Texas that were horrible, but you know, I bet they were crossbreeds. I bet they weren't. I've been in D. Lusby's yards, which everybody's always saying are Africanized honeybees, and if you go to the other end of the yard where she's not throwing boxes around and making them all mad, they're pretty normal bees, really. I don't know if she does that as just, I don't know if she just wants to test you to see if you'll stick around. I kind of suspect that. But I was in the back of the truck one day, and somebody in the truck said, well, I wonder how these bees would act if you just treated them normal. I said, well, let's go find out. So I took a smoker and we went to the other end, and we smoked them a little and opened them up, and they were just normal bees. And supposedly, you know, everything in Arizona is Africanized, and she does nothing but collect feral swarms to replenish things and open breed all of her bees in an area that they claim is totally Africanized. They're not that bad. Yeah.
You get into congested areas where we are, being so urban, and you really
Oh,
take a chance
you
with your
you can't afford to have mean bees in town. If I was in town and I had bees in this area, I would have a couple of things in place. One of them would be an escape plan. If I ever go out there and these bees are mean, I'm going to close them up, I'm going to load them up and take them to where? I should have that place already lined up, because if they're really mean and they're chasing people down the street right now, I need to do something right now, because that's not acceptable. Yeah, I think it's very important that if you're in town that you have gentle bees. But what I'm saying is, if you keep buying European bees and putting them in there, you're more likely to end up with mean bees. That's what I'm telling you.
I used to
Yeah.
do a sprinkler trick a couple of times on hives that started waterfalling on me with much success. And as long as the sprinklers stayed on and stuff, and things like that, worked great.
Well, you can't do that 24/7 though. But but that'll buy you a little time, yeah.
Yeah.
But I think you ought to have a plan because if you keep bees long enough, I don't care if you're in an Africanized bee area or not. If you keep bees long enough, you're going to get a mean hive sooner or later. And if it's in town, it's irresponsible to leave it there. It's just like keep having dogs. If you keep dogs your whole life, sooner or later you're going to get a dog that's mean. What are you going to do when you get a mean dog? Well, you're going to do something about it. You're not just going to let it bite the neighbors or run down the street. You're going to do something about it. You need to have a plan. Yeah.
I have a question, because we keep hearing about mean bees, mean bees. How would you describe a truly mean, bad hive, hot hive? What exactly is that? How would you describe it?
I admit I've been doing this a while, so maybe I have a better frame of reference. But anytime they're meaner than I want them to be, I put a red tack in there, because they can always have a bad day. And the next time I come back, if they do it again, I put another red tack in there. And when we get to 3, I requeen them.
When you say meaner than you want them to be, how is meaner than you want them to be? Some people have a high tolerance for mean, and some
Bees.
people don't.
The problem is, as a beginner, you may not understand what mean is. Bees in the air is not mean. That's just bees in the air. Bees in the air are bees in the air. Bees bouncing off my veil are being a little bit aggressive. Bees that are clumped on my arm and they're all trying to sting me, that's vicious. That's not just a little wound up. That's vicious. But yeah.
I kind of have found in my hives that if I have one where I pop the lid off, I smoke the entrance, pop the lid off and hit them with a smoker and they come out instead of going in, that's a pretty mean hive. And it's not, like you say, necessarily going to remain mean, and often it's just because maybe they're queenless or something.
Right.
But I've always found that, so far I've found that if I smoke a hive and the bees come out instead of going in, it's a mean hive. Those bees will follow me around the yard and half a block away.
You kind of got to set your own limits on what you're willing to tolerate. But I think anytime you have bees that are trying to sting people who are just minding their own business and aren't right in front of the hive, you need to do something, because it's just
They
irresponsible.
also release a pheromone
to the other
too that
people.
You can smell pretty strongly when they're attacking.
Right.
And if they constantly.
Oh yeah, if you're smelling that artificial banana smell, you guys remember you remember the banana candy you used to eat? It doesn't really smell like bananas, but it's supposed to smell like bananas. That's kind of what alarm pheromone smells like. So if you're smelling that strongly, as soon as you open the hive and they're just all over your veil, I'll guarantee you, if you can't see out of your veil, you got problems. Okay, you definitely have an issue. I would have a plan for where to put them if they've turned mean on me, and then you can deal a place you could put them where they're not going to hurt anybody, and you can deal with the problem, because it may be they're just queenless. It may be I've never killed a colony. I've always requeened them, but it's really difficult to requeen a big, booming, really strong hive that's really being mean without breaking it into little pieces and then requeening all the pieces, because it's just too hard. You can't find a queen in there. Yeah.
I had one last week that was real nasty, and I relocated it. First I tried to requeen it, and the queen came out and they killed her, and they made all new queen cells. I went in and scraped out all the queen cells and tried to put another queen in there, and I can't find her yet, so I think they may have killed her. And that's after I stacked them on top of another hive and broke them off.
That's one of the problems with an aggressive colony. Try a pushing cage for a introduction. You put the cage over some emerging brood, and put the queen in the cage, and she can start laying, and the emerging brood starts being her attendants, and she starts she starts laying some eggs, and she's a lot more acceptable to them at that point than when she's in a cage and she's not laying.
I figured my next step was just to break it up into like three nooks.
That's the other thing. The more you break them up, the more likely they'll accept a queen, and the more likely they won't be so aggressive. So
Right, it gives them that.
if they're really aggressive, that's my first thing is put a bottom board at the old location, put an empty box with some drawn comb there, and put a lid on it, and move all the rest of it, put every box on its own bottom board with its own lid, and then let them all settle down, because all the field bees will go back to the old location on that empty comb. And now I got rid of them, and I know the queen's not in there, and I got rid of all the field bees. And now I can start looking, and whatever these boxes is the lightest probably has the brood in it, and that's probably where the queen is, and I can start looking for the queen in there. And if you got queen cells, this is a wonderful thing about raising your own queens. If you got queen cells, you can basically just put a queen cell in all of those and come back in a couple of days and see what happened. If they tore down the queen cell, and put another queen cell in there and see what happens.
I
But
found that queen and I smashed her up all over the new queen cage.
yeah, I've heard of people doing that. I've never tried that. But anyway, yeah, I'd make splits. Let's get back to this, I guess. I think anybody ought to be thinking in terms of how many bees do I want to have in the spring, and what are my usual losses, and try to have enough bees going into winter to have
You might
that
wanna
much.
know, we really don't have much of a winter.
Well, that's true. That's true. That's the problem with traveling around the country too. Is some places I'm at, you know, they actually think they have worse winters than me, which they don't, and other places they don't have winter at all. So, yeah, you don't really have winter, but you must lose bees sometime or another, huh?
Yeah.
Off and on all year long.
Off and on all year long.
Is there any use of probiotics associated with keeping a good ecology?
I think the problem with probiotics is that we don't really even understand what should be in there and in what proportions. So I'm not so sure it's all that helpful. I think USDA is working on some research on that right now, but I don't know of any that's been published. And I don't know that I would trust it that much anyway. Somebody's been recently doing some research on what's living in bees, and they got bees. Europe actually has wild bees. I mean, literally wild bees. They've been living in the forests forever, and then, you know, what we have here we call feral bees, because the theory is that there weren't any bees here and that the colonists brought them all over. Even in 1926, in the American Bee Journal, they were still discussing whether or not that was true, and nobody's ever come up with any more or less evidence that there were or weren't native bees here. But the general consensus is that we assume that they weren't, and that these are all escapees, therefore they're feral. But over there, they've gathered some from over there because they figured these had not been interfered with by people or treated, and looked at the guts, what's living in the guts of those bees, and it's different than what's living in the domesticated bees, and it's different than what's living in the bees in this country that even haven't been treated for the last 5 years. They still don't have the rich ecology of those wild bees. I think the best way to inoculate bees with the right stuff would be to get bees that have the right stuff, and then put a frame of those bees in your colony. I think that would be the right way to inoculate them. I think trying to culture something in a petri dish and put it in there is probably not going to be successful, in my opinion, but that's my opinion. Because I think it's just way too complicated. I don't think you're ever going to be able to figure out what all the relationships are, but they're already established and living in these some of these wild ones. It's interesting. The person who was doing the study was contacted by Dean Stiglitz, and he convinced them to take some of D. Lusby's bees, which haven't been treated for a couple of decades now, and what was living in the gut of those bees was different than what was in all the commercial bees, and even in the bees that people had been not treating for 5 or 6 years. It was different, yeah.
Wouldn't there be possibilities too that there might be its own rich ecology, and where she is out in the mid,
True.
I mean, localized.
Hard to say.
The ecology of the bees in Europe is a different location, so maybe their ecology might not require what hers requires. I mean, there's temperature differences. There's all.
Oh, but
these things.
they're comparing not just with the European ones, but with what
Right.
everybody
I'm
who's
just
not treating
wondering.
here. But yeah, it's possible. But pretty much the environment in a bee colony tends to be fairly consistent, because they tend to control the humidity and the heat and all that kind of stuff. So I don't know. It's hard to say. And that could certainly have an effect. Yeah, I don't know. Yeah.
If there weren't bees in this country, How should pollination happen?
Well, that's a whole nother discussion. I could send you a whole article from 1926 in the American Bee Journal where several people present the argument that there were native bees, and the article right after that was Frank Pellet asking if anybody had any more evidence one way or the other, because there didn't seem to be enough evidence to establish this as a fact one way or the other. But the fact is, I don't think we know if there were or weren't. There were some native pollinators, though. There were alkali bees and squash bees, and there's a lot of native solitary bees, but and there were in South America there were some stingless bees that did pollination too, but they bite, but they don't sting, and they're small. They're really small, and they aren't real productive. I mean, you keep a colony of honeybees, you get 40 to 200 pounds of honey depending on the year, and you keep these and you might get a couple of pounds, So it was never very effective. But and as far as pollination of wild things, there's a lot of different pollinators. So the argument the scientists would present is that it was all these native pollinators that were pollinating what was here. The problem is we brought a lot of European plants in that those native pollinators wouldn't pollinate, and that's another reason we brought honeybees by their view. I don't know. I don't want to get into a discussion of native bees. That's a whole other discussion. Frankly, I think it's a fruitless discussion because I don't think you can prove it one way or another. So and they're here now. The Sierra Club's running around saying we should get rid of honeybees because they're not native now. I don't know if you knew that, but I find this an interesting prospect because there weren't any Europeans here either, but they don't seem to be trying to get rid of all the Europeans. And frankly, as a Native American, I don't think that's such a bad idea, but I don't think it's going to happen. So, well, it's like even half the plants you have out here aren't native, but they're here now and they're not going to go away. So, so what are the advantages to not treating? Well, you don't have to buy the treatments. You don't have to drive out to the yard and put them in. You don't have to drive out to the yard and pull them out. You don't have to contaminate your wax. You don't have to upset the ecology of the colony. You don't have, and you can breed for bees that can survive, and you can breed for mites that can live with the bees. And I don't think you can do any of those things until you stop treating.
Mike, we wanted to know, on your trip back home, did you want us to send home a case of small hive beetles with you?
Yeah.
No, we have small hive beetles in Nebraska. We don't have a problem with them, but we have them.
Oh, they're a problem here.
I've seen 3, I think. Yeah.
You do that within like maybe a half an inch.
Yeah.
Stuff, you know?
I think I had one mating nuke once that actually got slimed and I froze it, but no, I haven't seen very many small hive beetles.
Wait till you go out there.
No, I have seen them other places. I've been in North Carolina, and I never opened a hive in North Carolina, but what you didn't see 20 of them on the top right there as soon as you pop the top. Yeah, that was in a normal, healthy colony that had no problems, yeah. So, yeah, I've seen them. I think part of it is the winter and part of it is the clay soil. I don't think they reproduce very well. My soil right now, you couldn't dig it with a shovel. You'd have to have a pick, and it would break like rock. So I just don't think they succeed very well at reproducing in clay.
We don't freeze. We don't get the winter freeze, so the cycle hardly ever gets broken.
Right.
There's no freeze.
Small hive beetle's an African native, so it's not used to winter
Right.
either.
Well, in theory, they survive in winter climates, and they do seem to survive, but I think it kills a lot of them off.
In North Carolina, that's a winter
Yeah.
climate.
Yeah, in North Carolina, it's certainly, I mean, it freezes in the winter, yeah. It's not winters like I have where I am. I mean, we get minus 27 for a couple of weeks, so.
Come visit.
So my second step for 4 simple steps to healthier bees is to breed local survivors. Now this is, of course, we already touched on, and it's a controversy here, but I still think it's the right thing to do. I see the same thing in the Caribbean. There's a lot of Africanized honeybees in St. Croix and St. Thomas and all those places. And there are people who want to keep bringing in Italian stock because they think it's going to calm them down. But some of those bees are perfectly nice, and some of them are really mean, and you just need to breed from the nice ones. I don't think the solution is to keep bringing in stock and creating F1 hybrids that are even meaner, because you're going from mean to ferocious when you do that. You're not. I don't think that's the solution, but that's another. Local survivors are adapted to your climate. They're bred from survivors who can handle the challenges of your area, which might not be the same as the challenges in my area. Certainly where I am, I need bees that are hardy enough to get through winter and make the right decisions to get through winter. And those same decisions might not be the most productive here, you know. The other thing, if you raise your own queens, you can do it at the optimum time to get them really well bred. To me, a good queen, the most important thing about to get a good queen is that she's well fed. And the number 2 most important thing to get a good queen is that she's well bred, that she's bred with plenty of good, healthy drones. And the optimal time to do that, I don't know, maybe you have drones all year round, but I don't. So you can do it at an optimal time. Maybe if you do it in a
Year when we have more of them, less of them.
if you do it in a flow, when you have lots of drones, you're going to get well-fed, well-bred queens, and you can raise them and never have them caged. You can raise them in your mating nuke and then move them into your hives whenever you want, or you can actually put the cell in your hive and let it requeen your hive, and then you don't even have to find the queen. You just put the cell in, let her emerge, supersedure happens, you've requeened your hive, and you haven't even had to go look for the queen. But the fact is, once a queen starts to lay, her ovarials start to develop. And they keep growing and growing until you interrupt that, until they're done, or until you interrupt that. And when they're done growing is about 3 weeks after she starts to lay is when they're pretty much done. If you interrupt that at a week or 4 days, then her ovarials get stunted. You interrupt that by caging her and putting her in a cage and putting her in a queen bank, and now she's not laying, her ovarials don't finish developing, and she never amounts to as much of a queen. And she doesn't make as much pheromones, the bees don't accept her as well, and they usually supersede her. Typically where I am, you get a package and you put it in, and you can see it on the bee forums every year. People buy a package, they put it in, and immediately they're superseding the queen. And that's if they bothered to stick around and didn't move next door to the queen they liked better next door because that one was so poor. But I think that's because they get caged. As soon as they put the cells in the mating nukes, they come back and they open it up, and there's a queen in there. That's all they want to know. They put her in a cage and they put her in a package and they ship her to you. And meanwhile,
21-day period is very important.
and yeah, that first couple of weeks is the most critical, and 3 weeks is probably ideal. So after that, I think I've tried banking them. I used to think that they were bad because they banked them too long. So I tried getting some, letting them lay for about 3 weeks and banking them early in the spring, and I'd introduce them to a colony in the fall, and they'd start laying the next day, and they laid up a storm. So I don't think banking them is what hurts them. It's banking them too soon that is what hurts them. But you can make sure that never happens. Now, the point is, you can do this. The guy who's trying to raise queens for a living can't afford to do this. If he waited around for 3 weeks, you maybe have to do some queen rearing to really get a grasp of this, but it takes resources to set up all these mating nukes. And every one of these mating nukes, I could put a cell in and get another queen. And if I only leave her for a week, I've got 3 times as many queens as if I leave her for 3 weeks. So I got to get another cell in there so I can get another queen so I can ship it out the door because I got orders to fill. You know, I've sold all these queens already. I got to get them out the door, and I got a family to feed, and I got kids to send through college and whatever. Well, then there's just no time to do a good job. So one of the reasons that people can sell you queens for whatever the cheap price is right now, I don't keep up, but maybe $20 or something, is because they have to take shortcuts. They can't raise them for that. I don't see how a queen can only cost $20, frankly. You know, I know it seems outrageous, but to me, it isn't even worth the effort to try and raise one for $20. I got better things to do with my time. But on the other hand, the success of your colony depends on your queens, and you've got a stake in this. So the guy who's selling you the queen for $20 that's going to fail and get superseded doesn't really have a stake in it, because with any luck, you'll buy another queen. I'm not saying he's trying to rip you off. I'm just saying he doesn't have a stake in it. So you can raise better queens, is my opinion. The other thing is, 2 other issues here are longevity. They're trying to convince you should just buy a queen every year. Now you're talking every 6 months here. and requeen all the time. And that way you've always got a young, prolific queen. Well, the fact is what you have is a poorly mated queen that got banked too soon that you replaced the old one with that didn't have any longevity because she wasn't mated very well to start with. My queens tend to be 3 or 4 years old. They typically get replaced at about 3 years. Some of them 4 years. Kind of depends.
Not
But
by you, but by the...
no, not by me, by them. Yeah, but I used to mark them all the time. And I knew exactly how old they were by the color of the dot. So I know how old they were. I know if that was still the queen that I put in there 4 years ago or not. And typically they get replaced at about 3 years, and sometimes they make it to 4. But then again, I'm not putting chemicals in there, which all those chemicals affect the drones and whether they're fertile, and the queens and whether they're fertile, and the longevity of the queen, because the queen lives long enough that constant exposure to those chemicals is going to shorten her life. So.
Do the queens have like a peak time when they're better at
Well,
laying?
they probably do the best the second year, I'd say. If I was picking a year, I think they do the best. I think they do best their second year, but they do all right their first year, and they do all right their third year. But they're probably at their peak their second year. But part of the reason I don't requeen them is that if I keep requeening them, then I'm not selecting for bees that can sense that the queen's failing and replace her. So now I've made myself responsible for that. In the wild, any colony that can't sense that the queen's failing and replace her in time perishes. So there's a very strong selective pressure in the wild for bees to be able to sense that the queen's failing and replace her. But if all the beekeepers keep constantly replacing the queens, then there's no selective pressure for that. And pretty soon you have a bunch of bees that can't sense that the queen's failing and don't know to replace her. So I prefer to let them requeen themselves unless they seem to be failing to do it. And then I'll step in and put in a cell from one of my breeders, which are bees that know how to sense when the queen's failing. So hopefully I've fixed the problem.
When you have your yard set up as far as mating, do you have drone hives that are mainly drone hives?
The drones I want are the wild ones out there. That's the population I want. So, no, I don't.
So,
But then again, I don't make any effort to keep them from raising drones either. So, there's plenty of drones.
I don't make any effort to save mine from drones either. But, so if you're pulling an egg from your best hive, I've got, 3 hives that I have never removed. They're 4 years old, and they're, I'm sure, they're my most productive hives. And whatever they do is what they do. I never bother them in their brood chamber or anything like that. Sure, they've all swarmed every year for years, but they continue to produce, and they continue to make themselves new queens, I imagine. So from those hives, those should be the ones that I would choose to use to breed myself some queens.
Well, I'd say anytime you're trying to pick what you're going to use for your stock is pick stock that is what you want. Sure. What I want is healthy, productive, and gentle. If they're healthy, productive, and gentle, that's pretty much what I want. Everything else is kind of secondary. If they make a lot of propolis, they make a little propolis, I don't care. A lot of propolis I'd probably tend toward preferring just because I think it keeps the bees healthier. But all in all, I don't care if they're healthy and productive and gentle. That's what I want. Now, if they're swarmy in the sense that they tend to swarm and after swarm and after swarm and after swarm, that's probably not what I want.
Well, when that
If they-
happens, they generally die out.
I mean, exactly. Those kind of bees, they usually don't. Yeah, they usually swarm themselves to death, and then that's the end of that. But so there is such a thing as swarmy bees, but I don't see that bees swarming makes them swarmy. That's just how they reproduce. So, but on the other hand, I'd be trying to keep them from swarming because I can get a lot more honey if I keep them from swarming. But that's
By splitting.
well, maybe. Actually, I try to do that by not splitting. I try to do that by keeping the brood nest open, but that's a whole nother hour topic. But the other thing about keeping queens around, you'd be surprised how it changes your whole view of the world. I don't know how to explain that. I'll give an example. My dad bought a brand new van, and he was going to cut a hole in it to put a furnace in the floor of this van, right? And he's got a friend who runs a body shop. And my dad has spent, sleepless nights trying to figure out exactly where he wants to put this furnace. And so he goes to his friend, he says, I'm just not sure if I want to put it here, if I want to put it there. I'm afraid I'll put it there, and then I'll decide I didn't want it there. And the guy looks at him and he says, well, I don't understand what you're worried about. If you don't like it there, we'll just move it over. We'll just weld that old piece back in and cut another piece over there. And when you're a guy who runs a body shop, that's how you think. When you're a guy who's never cut a hole in your car before, you're thinking this is a really critical decision. Well, when you've got queens sitting in nukes, a lot of your decisions get really easy, It's like, should I buy a queen? Well, I've got a queen. Are they queenless? I don't know. Maybe they are, maybe they aren't. I could always just combine them with this nuke and not worry about it, right? It really does change your outlook on life to have queens, and queen cells are handy to have. Like I said, you want to requeen a hive and you can't find the queen, and you bought a queen, you got to find that queen before you introduce this queen or they're going to kill her. So what are you going to do? But if you got a queen cell, you just stick a queen cell in there and walk away. It doesn't matter. That queen will emerge. It'll supersede the other one. You've requeened them. You didn't have to buy a queen. You didn't have to worry about whether they were going to kill her, and you didn't have to find the old queen.
Is there a chance that young queen could get damaged in the fight killing the other queen?
It's pretty unlikely. She probably won't even fight the old queen. She'll just emerge, go out and mate, start laying, and they'll eventually get rid of the old queen.
Really?
I've seen where the old existing queen has gone up and torn the queen cell apart.
Sometimes.
Yeah, a lot of times.
I don't think it's the queen though. I think it's the workers. But
When you use a cell protector.
I never use a cell protector. But yeah,
Just a question on
I got a whole box of them. Never use them.
Just a question on the keeping the spare nukes for the queen. That sounds great and everything, but obviously they're laying. So what do you do? Just take the brood and combine it into your other hives? How do you keep a nuke just a nuke?
You do whatever's convenient for you for what it is your goals are and for what it is you're doing. If you can let it grow until it becomes a full-size hive, you can steal frames out of it to boost the one over here that's not doing so well. You can keep stealing brood out of it and try and stock this one up really well so that it'll hopefully make a big honey crop, and keep feeding combs in there to get them to draw more combs because you wanted more combs drawn. You can manipulate it any way you want, depending on what you want for an end result. A lot of times I just let that mating nuke fill up, and if I don't have a use for this queen, just put it in a bigger box, and then put it in a bigger box, and put it in a bigger box, and maybe it grows into a colony and maybe it doesn't, and I'm not. But meanwhile, I've got a queen if I want one, and other resources. But
So if you're manipulating that nuke, I've manipulated nukes to the point where they get upset that I'm manipulating it so much and they abscond.
yeah, well, that's your Africanized bees. Yeah, they Africanized bees are kind of that way. They're more likely to abscond on you because they don't like being messed with. Probably just move them into a bigger box and let them grow if you've got that issue. I think the most important thing about all this, though, is that we have a very limited gene pool here to start with. In theory, most of the bees that the commercial people are raising are descended from a few that got brought over from Italy in the 1800s, and a few that got brought over from Europe, the Carnies and the Caucasians back in that same time period. And really there hasn't been much new stock come in, and then the commercial people are raising hundreds of thousands of daughters, full daughters of this one queen, and selling them all over the country, and that really bottlenecks the gene pool. There are local feral survivors that are pockets of genetics all over this country, and if everybody would be raising their own queens, we could keep that genetics in the mix instead of constantly taking this very narrow pool and spreading it back out there so that it's crowding that out. I think that's important. I think that's important to the survival of the species. This is a quote from Randy Oliver: "If you're not part of the genetic solution of breeding mite-tolerant bees, you're part of the problem." I think this is an important concept to grasp, and that I hate to get into the them and us thing, but the fact is everybody who keeps bringing in outside stock constantly that can't survive varroa keeps watering down the stock that's trying to build up a resistance to it, So it's actually counterproductive to the people who are trying to get bees that can survive for people to keep bringing in bees that can't survive. And it's not contributing to the solution. This is applicable for your area. I'm amazed how often on BeeSource I see people that, you know, somebody says, well, I got this hive, it's really aggressive, and immediately 20 people tell them they got to get it tested to see if it's AHB. Well, if they're mean, they're mean. Are you going to keep them? I don't understand why you're having them tested, because I'm not keeping them if they're mean. I don't. And if they're not mean, then why would I test them? I mean, there's no reason to test them.